Okay, here we go with the next set of notes in our Chemistry of Life series, this one on lipids and nucleic acids. On to lipids. Lipids are just fats, oils, and waxes, more commonly known. Their monomer is just simply called a fatty acid. And these guys are mostly used for energy storage, so think of the fat that you might put on as being long-term energy storage, but it's also a major part of cell membranes and also a little bit of part of cell-cell communication, so how cells talk to each other or maybe recognize each other. Saturated versus unsaturated fats, something you might have heard of before. Saturated fats are the fats that are solid at room temperature, generally speaking. These are found in meats and dairies, like the fat that would be on the side of your steak or in your bacon, the milk fat, things like that. They're generally more solid at room temperature, like cheese. Unsaturated fats are just the opposite of that, right? They are liquid at room temperature, so think of olive oil or canola oil, stuff like that. The reason that is because of the chemistry of the molecule, as you might have guessed. Saturated fats are one long chain like this. You got lots of single bonds in there. You have one long, narrow chain. So because they're long and narrow, these chains can pack in nice and close to each other, so that fat becomes very, very dense. So think of the fat on your bacon. Very, very, very dense. Yes, it can turn to liquid when you add heat, but what happens to your pan about an hour after you cook bacon if you don't clean up right away? You come back and you got globs of fat in there. So that's a saturated fat. These guys are very close together, like lots of people packed into an elevator. Unsaturated fats, however, have this double bond. And as a consequence, they have an empty spot here and an empty spot there where there's no hydrogen. So these guys are not saturated with hydrogen. Every carbon molecule or carbon atom up here is attached to a hydrogen. It is saturated. It is full of hydrogen. This one is not. And this also gives it a little bit of a bend. So we can't pack these molecules in as tightly, so they're less dense, so they stay liquid. This would be like that same elevator. If we all went into the elevator sticking one arm out straight, would it be able to fit as many people in there? No. It would be able to move around a little bit more within that elevator. So the main use of lipids, like I said, is pretty much energy. If you look at it, something like glucose, we said is quick energy. You get energy when you break these carbon-hydrogen bonds. It's a very easy bond to break. That releases energy. You've got a fair few carbon-hydrogen bonds, right? But not nearly as many as a lipid might have. So it's got lots of those. They're very, very densely packed in there, though, so they don't get released as easily. So it's not a quick source of energy. This energy over here is going to be very quick, right? This over here is going to be more like a long-term storage. So your spare tire that you might have, long-term energy storage. Like I said, it's also used in the cell membrane, the phospholipid bilayer, so lipid bilayer. We're going to talk about that a little bit more later on this year. And then cell communication, so things like cholesterol. You might think of cholesterol as a bad thing, something you're trying to avoid, but cholesterol is something you actually do need some of. Too much is a bad thing, but it helps cells communicate with each other. Finally, we move on to the last of our 4 organic macromolecules, the nucleic acids. So nucleic acids are made up of monomers called nucleotides. You guys have this diagram in your sheet. I would recommend attaching it here. And a nucleotide is a phosphate, a sugar, and a nitrogen base. So this right here would be one nucleotide. So one nucleotide right here. Right? You've got your phosphate, which is this little square or circle right there, the sugar, which is this 5-sided pentagon there, and then the nitrogen base. So that's what makes up a nucleic acid. And then obviously nucleic acids mostly would be considered, or a good example would be DNA, but there's also RNA, which is like DNA's little cousin that we'll talk about a little bit. A little bit more on DNA. DNA stands for deoxyribonucleic acid, so the Na is right there in the name. Deoxyribo comes from the type of sugar that it's made of, deoxyribose. And so again, we see that nucleotide. You've got your phosphate, you've got your deoxyribose sugar, and you've got that base. And DNA has the characteristic double helix shape. That's the most important thing, having that double helix. within that DNA, within the nitrogen base that you just saw, you have 4 different options of nitrogen bases. Right? There's 4 bases in DNA. There's the pyrimidines, which are a chemical called cytosine or a chemical called thymine. Right? These are right here, cytosine and thymine. And these guys are a single ring structure. Okay. There's also another type of pyrimidine called uracil, which we're not going to worry about for DNA. Right? Not in DNA, it's only going to be in RNA, which we'll see a little bit later. Then your other option, of course, being the purines, which are adenine and guanine. Adenine and guanine, and these guys are both double rings. Right, so those are those 4 different options of things that can go in this base segment. All the other nucleotides are always going to have the phosphate, they're always going to have the deoxyribose, but this base thing can either be a C, a T, an A, or a G. So all this stuff goes together to form that double helix shape. The DNA double helix is made up of a sugar and a phosphate backbone. So you can see here the phosphate and you got the sugar. Those guys are all connected together, making up the backbone of the DNA. And then the bases are like the rungs of the ladder. So you got the bases on the inside there, as if you were to uncoil it, it would look like a ladder. Really, really super important thing is the way that these bases pair together. They pair in what are called complementary pairs. They complement each other. A always pairs with T, adenine always pairs with thymine, and cytosine always pairs with guanine. And that has to do with their chemical structure. If you look, you got thymine in red right here, and adenine in blue. You got a single ring paired with a double ring. Single ring paired with a double ring. Single, double. Single, double. Single, double. You never have a double and a double, right? That would be like having a ladder where some of the rungs are 1 foot apart and some of the rungs are 2 feet apart. That just wouldn't work. So you have to have a single paired with a double. And if you'll notice that between these two, the A and the T, you always have 2 hydrogen bonds, versus the C and the G, you have 3 hydrogen bonds. So the way they work, it always chemically makes sense for them to go A paired with T, A paired with T, C paired with G. Always, always, always. When you talk about DNA, A pairs with T, C pairs with G. The structure of DNA was actually only discovered relatively recently. We've known about DNA since like the 1800s. I think that's when DNA was first isolated. But in the 40s and 50s, there was kind of a race going on to try to figure out what DNA actually looked like. And it was basically first discovered by 2 guys named James Watson and Francis Crick in 1953. So they're the ones that get all the credit for it. But kind of one of the curious stories in biology, or in science, I guess, in general, is they actually had a lot of help from this woman here, Rosalind Franklin. And she had taken these x-ray pictures, like this guy here, that kind of gave them the clue, kind of said, oh, hey, wait a minute, what if we try a double helix shape? And unfortunately, as was a lot of the case in those times, the women in science weren't given all their credit, and when it came time to give out the Nobel Prize, she wasn't given any credit at all. She'd actually died by then. She had gotten cancer probably from working with x-rays so much, they didn't really know a lot about them. They didn't realize they caused cancer back in that day. So kind of an unfortunate story, but she's definitely more widely recognized for getting credit for that now. In fact, she was just given a Google Doodle on July 25th, 2013 in honor of what would have been her 93rd birthday, in honor of her contribution to science, which I think is kind of cool that she's being recognized a little bit more nowadays. And then in addition to DNA, there's also RNA, which stands for ribonucleic acid, because just like DNA, it's made of sugar, which is called ribose instead of deoxyribose, and then the phosphate backbone, so phosphate and ribose. And then it has pretty much the same bases with a couple little changes, as adenine and guanine and cytosine, just like DNA does, but instead of thymine, it has a base called uracil. So similar to DNA, C still pairs with G, but in this case, an A on DNA is going to pair with a U on RNA. So anytime there's an A on DNA, it's going to pair with a U. And you see over here you've got U's instead of T's. And that pretty much wraps up our tour of the chemistry of life, the macromolecules and water, and how all that works to help keep our bodies going.